This video is about what kind of results one can expect when you take DNA tests. So to summarize it in most succinct way, there are three types of results you can expect when you take the DNA test. Number one is that you will receive negative results, which means nothing of concern was found. The alternative to that is that positive results were discovered, which means further investigation is warranted or medical management has to take place because something was discovered. This might seem counterintuitive to what you would normally expect because normally we associate the word negative with negative outcomes and the word positive with positive outcomes, but perhaps one way to think about it is that this answers the question, did we find anything in our test? And the answer to the question is, we didn't find anything, so the answer is negative, or yes, we did find something in a test and the answer is positive. The final potential outcome is that there is no results, and in this case it would mean that the test simply didn't work. So to summarize once again, negative results would mean that the person is presumed to be healthy as nothing was found. With positive results, medical management is expected. With no result, the only alternative left for you is to redo the test and provide the sample once more. What is important to add here is that the outcomes that are either negative or positive is usually what is only taken into consideration by the companies when they determine the accuracy of the test. And this is how companies can cheat a little bit by inflating the quality of these numbers by simply discounting the no results. And as you will see right away, no result can actually be not insignificant, and therefore this is one way of potentially skewing the numbers. Right before we get into further details of what no results mean, let me also expand on what kind of DNA tests we could be performing. And we can divide them into two categories. Number one is what we refer to as germline tests, which analyzes the DNA that you were born with or the DNA that you inherited from your mom and dad. And typically these type of tests do not fail often. That's not the determining factor of the quality. In this type of tests, usually the determining quality is how the data is subsequently interpreted. But there are other types of tests that do not look at your DNA you were born with, but additional DNA that might be found in your body. So we refer to these as non-germline, and one example is the non-invasive prenatal test that we use to determine the fetal DNA that is floating in maternal blood. What is actually being looked at in these type of tests is placental DNA, but since placenta and fetus are developed from the same fertilized egg, we use the placental DNA floating in mother's blood to study fetus DNA. So we're looking at completely independent individual here. The other possibility is to look at the cancer DNA. Cancer DNA evolves from your own germline DNA, but it mutates itself, and therefore it's as if you're looking at another individual. So in this case, we have to look at cancer biopsy, and it's like looking at cancer DNA within the sea of your own normal cell DNA. And in non-germline cells, there can be quite a difference in how frequently no result can be obtained. So at least with an example of the NIPT test, it can range from 0.1% to 5% of the time, and sometimes even more, of how frequently these tests will fail. So you can see this is about 50-fold difference between different types of tests. So no tests are created equal, and then there can be large variation in the quality of these tests based on how frequently they fail. So you can see that all these tests might report similar accuracy, but the reality is that the accuracy will not be the same because one test might fail much more frequently than the other. So selection of your test is very important. So why these tests will fail and produce no result? Most frequently, it will be because there is simply not enough DNA obtained in the sample. The other reason is that tests can simply fail for a variety of technical reasons. Now that we covered the effects of no results, let's look at the negative results in more detail. What is important to understand with negative result outcome is that it does not mean that there is no mutation found in your DNA. It all depends on how much information was actually being analyzed. So to give you an example, tests can be divided into three types of categories. Number one are tests that look at simply at very specific mutations within your DNA. So they only look at that mutation of interest. This is in contrast to tests that could be looking at specific genes that were already pre-selected for analysis depending on the type of condition being investigated. And in this case, entire genes are being usually investigated. And this in turn can be contrasted with looking at your entire genome, where all of your DNA is being investigated. And thus you can appreciate that the less information you investigate, the more likely you're going to encounter false negatives. What does false negative mean? False negatives means that you are told that no result was found, which to a client or patient might mean, hey, everything is fine, when in reality, something is actually found in your DNA or is present in your DNA, it just simply wasn't looked at. And therefore, you can appreciate that the more information that is going to be investigated within your DNA, the more likely you are going to discover a potential mutation. So to visually present that, let's pretend that this stretch of DNA at the bottom of the slide is an entire gene we are looking at. And limited tests that look at only at specific point mutations and nothing else could uncover these mutations if you do have them. However, within a specific gene, there might be literally hundreds of more other possibilities of mutations that have clinical outcomes that otherwise limited tests will not even look at and would never be uncovered unless you look at the entire gene. We will conclude the video now with looking at the most important aspect of DNA testing results, and that is the positive outcomes. If you order a type of DNA test that is a clinical DNA test, you can expect that a medical report will be issued to the ordering doctor. So within this type of test, there's a variety of different results that could be discovered. Number one are pathogenic mutations. These type of DNA mutations are ones where there is a strong evidence already established that the DNA mutation leads to a specific disease or a specific medical condition of interest. You could also have a likely pathogenic DNA mutations, and these type of mutations are also important to report, but they might not have as much collected evidence as the pathogenic ones. Nevertheless, both of these type of DNA test results will always be reported to the ordering doctor, and these type of results are indeed used by the doctor to determine future medical management of the patient. Next alternative are variants of unknown significance, or VUS. These are the type of DNA mutations that we simply do not know what they do, and therefore they are almost never reported to a doctor. Occasionally, they might be reported to a doctor if they're very suspicious for a variety of reasons. For example, there might be linked to the patient outcomes or family history, but nevertheless, variants of unknown significance are never used by the doctor for medical management. If they are ever reported, they will be reported only for tracking purposes, meaning we want to keep an eye on these in case as we gain more information about genetics or we sequence more and more people, variant of unknown significance might be reclassified to a pathogenic mutation. That would be the only reason why they're ever reported. The final two alternatives are DNA mutations that are already known to be benign or likely benign, and these will never be reported to a doctor. And the reason why is simply because both benign DNA mutations as well as VUSs can number literally in tens of thousands within at least a human genome. The final note to add before we conclude the video is a special consideration for the results, positive results of non-invasive prenatal testing. And the reason why is because NIPT tests, they do not look for pathogenic mutations within the DNA. They might sequence the DNA, but what is actually being measured is the amount of DNA discovered. And in this case, we want to see the difference whether the fetus has a normal amount of chromosomes versus a positive result where we might discover an extra chromosome or potentially a missing type of chromosome. So this is a distinction that needs to be made for NIPT test, that we are looking at the quantity of DNA and not the actual sequence DNA mutations. And this concludes our video about what kind of DNA test results you can obtain. And we're looking forward to seeing you next time.